guess about what molecule in the cell is making all of this stuff happen? Moving the chromosomes around, getting stuff done. What would your bet be on?
Enzyme.
Some kind of protein, right? That's doing this. And so indeed, there are a bunch of proteins that move these things all around, just as there are proteins that move in your arms to let your muscles contract and your joints move. So there's fibers of protein that are able to move the chromosomes around within the cell. And they all have names and complex molecular stuff going on, but we don't need to worry too much about that.
But is an enzyme a protein?
An enzyme is a protein, but not all proteins are enzymes.
Okay.
So the next step is we're going to take those proteins and we're going to pull on both of the sister chromatids in opposite directions. And as we do that, they'll dissociate from each other and move towards the opposite pole of the cell. So what will have happen here is the black chromosome will be pulled towards this pole, the blue chromosome will be pulled this way, and the same occurs on the other side. So we'll end up with a cell that kind of looks like this. When you see the chromosome sort of being dragged across the cell, right? Like if you grab somebody by the belt and pulled on them, they kind of fold at the waist as they're pulled along. You've entered into this phase that's called anaphase. Next, and it's actually, we're almost done. I mean, if you notice, we really have accomplished our second goal, which is what? Separate the copy, right? So we then enter into what's called telophase, and then, which is the final phase of mitosis, and it transitions directly into cytokinesis, which is step 3, the actual division physically of the cell. So you'll end up with a cell that has one black one on this side, one black one on this side, one blue one, and one blue one. And then the cell will begin to kind of pinch down, as you saw in the video, and get ready to divide. So when you see this starting to happen, and you even see the chromosomes in an actual cell not becoming as visible, or becoming less visible rather, you've entered into what's called telophase. And then the cells split, giving you 2 cells that each have, I hope we can all agree, complete set of all the information. So we went from 1 diploid 2n equals 2 cell to 2 diploid 2n equals 2 cells. Yeah.
And is it after this phase that they will reconnect then with another strand?
So what would happen next is if these were cells that were continuing the process of division, they would enter into the S phase, where they would synthesize a new complementary copy of each, and then proceed into mitosis.
Okay, so aren't you required, no matter what, to have 2 copies of a chromosome?
Well, they already do have 2 copies, right? They have 2 versions of the chromosome. You don't need 2 copies to be a functioning cell.
I see. Okay. I've just pieced together something in my mind that wasn't pieced before.
So this is the reason it's confusing, is because, yes, there are only 2 bits of information, chromosome from mom, chromosome from dad, but then you make copies of each one of them. And so keeping track of those gets really confusing as you move on. So what you guys are going to do is sketch out cells, and I'm going to give you a hint about how I like to approach the sketches. I drew it up here with the 2 different chromosomes, the homologous chromosomes rather, being different colors. But I think for you, a superior way to do it would be if, let's say you have a 2n equals 6 cell. What I would recommend you do is you choose a color to represent a given chromosome, and then either have a filled-in version or an open version to represent the 2 homologous versions. So for instance, you might have a red chromosome. This would be mom's version and dad's version. And then you would have a black chromosome. Mom's version and dad's version. And then you have a blue chromosome, mom's version and dad's version. So because we have so many colors, I think it's easier to just represent the homologous ones with open or closed as opposed to different colors.
The 2n equals whatever number. It keeps kind of confusing me.
Yeah.
So, what exactly does the 2n mean? It's just
2n refers to the one that it's a diploid cell.
Okay.
And then it tells you how many pieces the genome basically is broken into. So this is, if you divide both sides by 2, if you wanted to think of it that way,
Right.
you have n equals 3. So there's 3 fundamentally different sets of information. But you get 2 versions, one from mom and one from dad.
Okay.
So the only other thing that I might add is there are a couple questions in the beginning, the pre-lab section even, that ask you to sketch stuff out for meiosis. We haven't talked about meiosis yet. Don't worry about them for today. Only do the questions that relate to mitosis. All right? And if it asks you anything about before or after S phase, this is what it's referring to. And do any of them ask about G1 and G2? I can't remember.
Yeah.
Okay, good. They do?
Yeah, there is one.
Okay.
At least there wasn't a pre-lab quiz.
So, I'm glad that I asked that question. So, when a cell divides, once a cell has divided, and it's prior to the S phase where it's doing DNA synthesis, we refer to that period of time as the first gap, or G1. So G1, it's like, there's so much terminology, it's kind of insane. But G1 refers to the cell prior to synthesizing a copy of its chromosomes. After the synthesis of the chromosomes, it enters into G2 before it starts with mitosis. So if it says sketch a cell in G2, it just means after DNA synthesis. If it says sketch a cell in G1, it means before DNA synthesis. That's the only thing it's saying. And it's just using terminology to confuse you. And since I was involved in writing the lab manual, I can't really defend that. Okay? So go ahead and get started. And